Hello, I am Carlo Colantuoni, and I'm going to tell you about the work in our most recent manuscript that is here on bioRxiv. Shreyash Sonthalia is the first author, and it'll be out end of 2025 or beginning 2026. But I'll walk you through some of the things we have in that manuscript. As the title here implies, a big component of what we're doing is bringing together all of the transcriptional data in the public domain focused on neocortical development. The second piece of all of this that's built on top of that data collection is what to do with all the data. And our approach is a little bit different than most of the mainstream approaches going on in genomics or transcriptomics today. We seek to leverage all these different public data sets, but without pushing them into a common space. So it's a little bit different than the integration methods that are often used, and it allows each data set to kind of live in its own space. Um, and the three we're picturing here are a mouse, monkey, and human study, each mid-gestation, neocortex from the neural stem cells at the bottom of each of these maps to the IPCs, newborn neurons, and maturing neurons there. And we're going to focus in this short talk on studying the progenitor cells in this manuscript. Another whole half of it is looking at the birth and maturation of neurons, but here we're going to look at the precursors. And at the end, we'll hit this NeMO analytics site where all the data is assembled, and anybody that knows how to type on a keyboard can interrogate genes across tens of data sets from single cell, sorted bulk data, laser microdissection, um, and bulk RNA sequencing as well. I'll show you that at the end. Okay, so let's jump right in. These are the same three data sets, now colored by 3 of 7 of the pieces we decomposed. And I'll go over the decomposition in another short video on the same site. Check it out. But here we're showing the radial glial signature at the bottom, IPCs here in green, and then neurons in blue. But I want to stress that this isn't cell typing. These are the cells where these signals are highest, and it's sort of a complement to the non-overlapping cell classes that we often use in single cell data. Whereas here with these decompositions across matrices, we're looking for continuous signals that are enriched in certain cells, but that change as gradients of different levels across these cell transitions. There's not a step function, there's curves. So we're going to get into both the evolution and developmental dynamics of these transcriptomic programs in different neural progenitor cells across the mammalian neocortex. So again, we're not going to be down here spending all our time in the red here today in these radioglial cells, the neural stem cells of the neocortex. So this is that pattern just visualized again. It's a transcriptomic program that is really high in radioglial cells and especially in cycling cells. And the genes, the top genes in this pattern are sort of a hit list of radioglial markers, pleiotrophin, the Hess genes, and so on. Super enriched in cell cycle, this pattern is. So the gene weights underlying the picture that we're looking at here. And also, it's interesting, if you look at high penetrance, low frequency variants that cause disease, um, diseases that really disrupt the growth structure of the cortex are enriched. Microcephaly and hydrocephaly are really the genes responsible for these disorders are very highly ranked in this radioglial signature. Polygenic GWAS signals summarized via magma have that very little signal, but these have much higher signals in the neuronal patterns. But today we're focusing on these progenitors. So here are all of these values, the strength of this radioglial transcriptomic signature in all of the different cell types, and it's spread across age in each of these systems. So the coloring here is by NPC, dividing NPC, IPC, and then a glutamatergic neuron. And they're separated by those four classes in the same colors here, and they're spread out across the ages, 12 to 18 days in the mouse, about 50 to 110 days gestation in the monkey, and gestation week 16 to 24 in the human. This is mid-gestational peak neurogenesis in the cortex for each one of these species. And as you can see, there's these smooth um, age-related descent of the radioglial signature in both dividing and non-dividing progenitors, a little bit higher in the dividing ones, but over time, those 6 days in the mouse here, it's really descending. You can see it a little bit in the monkey. But there's also a little bit of this signal in IPCs and even neurons that's just being shut down here a little, even though we're already neurons, these programs are still going on. And this is the same kind of thing painted on top of pseudo time here. So this radioglial signature is very high, very early, and especially in the dividing cells, and then really comes down through the IPC stage and is low in neurons. Okay, so now we're going to look at the strength of this same signal, this radioglial signal, in different data sets to get more information about it. The first one here is just kind of a validation, another single cell data set in human where the signal is localized to the dividing and non-dividing cells, neuroprogenitor cells. Here, where they coupled laser microdissection with RNA sequencing, we can see it's localized to the ventricular zone where we would expect these dividing radioglia to be. The next study here sort of takes that spatial notion and really explodes it. This is much newer data from Chen et al. That has 30,000 gene expression values for every pixel in this image here, where we can see very clearly lining the ventricle both in the dorsal pallium and in the ventral ganglionic eminence, where the GABAergic cells are made. This program is lighting up all of those ventricular radioglia, and even in the midbrain and hindbrain, where most of these progenitors have already turned into neurons. There's a little bit left around the ventricles, and so we can really see in great detail where these cells came from, even though we captured the signals from single cell data that had no positional information. But we can map it via the genes onto these spatial data sets. The next example here is instead of space, we're now looking at time. So this is some flash tag data from Denise Jabaudon's lab. And so they record the time between the neural progenitor's terminal division and when it was sampled, when it was sequenced. So this neural progenitor radioglial signature is high 1 hour after that final division as the progenitor is becoming a neuron, but by 24 hours and then 4 days later, this signature is at its lowest and staying down there. And these other two are actually bulk RNA sequencing here from human, and then a spatial transcriptomics from the human. It's really high up very early in this is about week 13 here. So we really need better resolution early on to see when it peaks. But all the way 30 years later, a portion of this radioglial signature is still present. Here it maps to the white matter, where lots of OPCs likely, and perhaps astrocytes as well, are still expressing a portion of the neural stem cell transcriptome. That's been seen over and over and again in both astrocytes and OPCs, that they have a good bit of that neural progenitor signal left in their transcriptome. So now, again, looking at more data sets to simply get more perspectives on where and when these signatures appear. These are 2 laser capture microdissection studies from the Allen Institute that were coupled to microarray gene expression analysis. And so each single dot here is a laser capture from one region at one time of either the macaque or human development. And so high in black here is the ventricular zone. You can see it's super high and descending. Here also in the human, very high at weeks 15 and 16, coming down a little more by week 21. What you can also see is these light purple dots are about the same height as that black ventricular zone, that is the same thing. It's the ganglionic eminence, or the ventricular zone of the ganglionic eminence, where the GABAergic progenitors are. And as we saw here in the Chen data and spatial, the both dorsal and ventral progenitors of the telencephalon really light up with the signature. And all the green down here are differing chunks of the cortical plate where it's off in those terminally differentiated neurons in both the monkey and the human there. So here's one more view of some of this spatial data, just to show you the kind of detail. This was actually one time point of many in the Chen study. So that was the 16.5 time point. They had even back further, but what I'm showing here is just some sort of grand perspective. If you zoom out at E11 and a half, the forebrain, the telencephalon, is almost entirely still made up of progenitors, really on fire with the signature. In the hindbrain, you have or hindbrain or midbrain probably here. You still have a good bit of it, but you have a lot of neurons that have formed that are negative for it. But again, the telencephalon is a little bit behind. And so as you go along here, by 16, you have a lot of cortical plate that is negative for this progenitor signature. And kind of you can see the wonderful detail in between of this shift, where at these early times, there's almost no neurons in the telencephalon yet. So we don't have any negative cells, those light yellow ones yet. This is another really excellent data set that we collected for this study that is not only spatial RNA sequencing, but coupled to histone marks. This is visualizing the RNA sequencing, but in the paper, we go into some depth on looking at epigenetics coupled to some of these signatures. But you can really see throughout the brain, even the eye, and then this is probably spinal cord all the way down here. This radioglial signature really does track to the edges of the ventricle throughout the CNS. Okay, so that was kind of a low resolution decomposition. The pattern we were looking at was one of 7. So that means 7 transcriptome programs whose mission it was to explain all of the dynamics in neocortical neurogenesis across these 3 species. We're trying to pick out signals that are common across there. And so what we did is then go from this kind of low coarse resolution decomposition using the same 3 data sets, ask now for 40 instead of 7 patterns. And similar to how you can look at the bang for your buck in a PCA analysis where you look at a scree plot and how much variance you're explaining, what we do here is we run that same joint decomposition that we call joint NMF. It's using a non-negative matrix factorization, but the 7 is here, where the y-axis is how much error, how well those 7 patterns can describe the whole data set. And every time you add patterns, more and more patterns here on the x-axis, you can estimate that data set better and the error goes down. So with more dimensions, you can explain more, but there's some decreasing returns here. So we picked 40 as a level where we're getting a lot more granularity and still pretty good bang for our buck on increasing patterns. So here are 7 of those 40 patterns. On the right here, we have 4 different neuronal patterns. You can see just groups of neurons with differing signals at different times, maybe a little earlier, a little later, different points on these. Back here in the progenitors, we also get increased resolution. We have G2M and S phase specific signatures. Particularly in this macaque data, it's really beautiful. You can see the G2M phase are the cells on the outside of these loops. This is the IPC loop of cell cycle. This is the radioglial loop here. So on the outside of these loops is G2M. On the inside of these 2 loops is S phase. And here's another progenitor signal that isn't about cell cycle, isn't about IPCs, but is about radioglia and a particular slice of radioglia, as we'll see in a moment here. I gave it away a little bit with the label there, but we'll see why I labeled it ORG in a second. These are the top genes in this pattern that include pleiotrophin and a PTPR zeta 1, which is the ligand receptor pair that in Alex Pollan's original molecular description of outer radioglia in 2015 were described as markers. HOPX is another one. This FAM107A, I think there's HOXD is another one. or MOXD is another one, all very highly ranked in this pattern. Now, what's interesting is there's a very distinct population in both the macaque and the human for this, but in the mouse, the progenitors also are higher, but really the highest ones on the edge here are the only ones that really blazing. It's not as clear as it is in the mouse and monkey, and the primate and human enrichment of this cell type are consistent with that. So we'll get into that a little bit more here. So this is that same visualization. Now these little insets I've added here show the distribution of values. So basically the distribution of the color values in each of these plots, where the arrows are indicating in both the human and the monkey, you can really see very small but very skewed, very high values in a small number of cells, whereas in the mouse, we have kind of a shoulder coming off the main, low distribution, but no real big lump at the end. And what does that mean? Um, so what we looked at is the author's original cell type calls and the strength of this um, particular radial glial signature in the different calls. So I know these are a little small, so I'll tell you what they are. This is radial glia, intermediate progenitors, and then neurons in the mouse. So as a broad category, the radial glia is where this cell or this pattern is highest. We have a little more information in the monkey and the human because the cell type calls are a little more granular. In the highest category in both the monkey and the human, this first box plot is outer radial glia. The next one is truncated radial glia, and then otherwise unclassified radioglia, ependymal cells, IPCs, and excitatory neurons here. It is almost often. So really the highest in OPCs, not specific to it. You still have an elevated levels in other cell types, but definitely heavily outer radially enriched. Now this shows those marker genes I was talking about for outer radioglia. They're shown here on the x-axis for how highly they're ranked in this pattern. So that's why they're all to the right. But as you can see in the mouse, only about half of them, the ligand receptor pair, HOPX, tenacin C, are high, but the two or three other ones are almost off. In the monkey, they're all, none of them are off, but a couple of them are still low. The four that are high in the mouse are still very high or even higher in the monkey, and these couple are lifting off, and in the human, they're all up there. So these genes, it really looks like a transcriptomic program that is formed or coalesced over evolutionary time. Even in the scatter of things here, you can see it's increasing in the mouse. Again, perhaps corresponding to the increasing cohesiveness of this transcriptomic program over evolutionary time. So one of the things we noticed back here, before I get to that last panel, in the mouse, there's a whole bunch of genes, and I didn't really explain what the y-axis is. So the y-axis is the expression of these genes in cells that look like outer radioglial cells that have this pattern 27 in them. There's none of those in the human. There's really no genes that are expressed at high levels in these cells that have low levels in the x axis, which is their value in the pattern. So if you're in the pattern and you're in these cells in the human, you're high, and if you're not in this pattern, you're off in these cells. But in the mouse, there's these genes that are not in the pattern but are in these cells. And again, this contributes to our view of this program as something that over evolutionary time has become more cohesive. Notably, one of the genes that is high in expression but low ranked in this pattern, and specifically in the mouse, this is FOXN3 here in green, that is off in the mouse, or excuse me, the monkey and the human, but is on in the mouse. It's a transcriptional repressor and a proliferation inhibitor. And we loaded up the Trevino data, this human data down here, into Cell Oracle and did some in silico perturbations of the FOXN3 gene. And if you do that, if you have a computational knockout of the FOXN3 gene, analyzing the gene regulatory network, the Cell Oracle tool predicted that you would get 64% more outer radioglia than what is there to start with. And conversely, if you overexpress it computationally, you get a decrease of almost 80%. You end up with only 20% of the outer radiomedia that you otherwise had there. So um, this could be part of the regulatory network that is been emerging in the primate and specifically in the human to make this late radioglial state highly proliferative and increasing its neurogenic capacity. And FOXN3 could be part of that. So now, just like we looked at the general early radioglial pattern in lots of other data sets to understand what it was, we're going to do the same with this outer radioglial pattern. And what this is those same two laser microdissection microarray studies we looked at earlier. And again, the black is ventricular zone here. So when we look at this outer radioglial signature, why the heck would it be the highest in the ventricular zone? And it's not only high in the ventricular zone, but specifically early in development, it's high. And by later times, the ventricular zone in black actually ends up right where the inner and outer subventricular zone are, both here in the human and up here in the monkey, where the black is the highest, but by these later times, they kind of converge so that they're the same. But if this really is a signature of the outer radioglial cell, why is it the highest in the ventricular zone? So I pulled up Alex Pollan's paper from 2015 to look at this more. And this is a picture. They did um, some staining here at GW18 for what they found to be ventricular radioglial markers or outer radioglial markers. And they're super clean right along the ventricle with these genes. Way up here, clearly in this more diffuse, spread out outer ventricular zone for these poster child genes for the outer radioglia. So why are we seeing all this ventricular signal for something we think is an outer radioglial signature? Um, so here is another view of things, but now with some expanded time from Alex's deeper figures in this paper. So what he noticed is even though here at GW18, all of these genes are in fact markers of the outer cells in the outer subventricular zone. If you look back in time, this is 4 of those genes, and along the bottom we're going from about gestational week 13 to 20. You can see that each of these genes starts most highly expressed at the subventricular zone and perhaps the inner subventricular zone. So the ventricular zone down at the bottom, and then it just climbs up over time, up over time, up over time in each case. So that really explains this finding here that at early times, and this really corresponds to, right, this is 13, 14, 15, 16, and then by 18, 20, it's really high. Here is 15 and 16 weeks, and then down by 21 weeks, the ventricular zone is no longer where it's hottest. So again, being able to assess these signals in broad-ranging data sets in the public domain really give us the ability to understand what we're looking at in these signals. So now we're going to get even deeper into this exact same signature by looking at additional data sets. This is another mouse data set from Pablo Arlota's lab. The cortical development study across ages here from 10 to E10 to EP14. The radial glial signature we discovered first is very high in all of these neurogenic radial glial cells down here. But if we look at the one that's associated with the outer subventricular zone at later times, in the mouse, it is most high not in any of the neurogenic radial glia over here, but here in the gliogenic progenitors as well as the glia themselves. Now, this is very interesting because if we then look at the macaque and human data sets, the outer radioglial signature is not limited to the gliogenic progenitors as it is in the mouse. So here in the monkey, it's the heaviest. This is actually what's been called astrocytes over here, but it still has some pretty good representation in the neurogenic progenitors, which are here. Now that's where the earlier signature for radioglia is highest. And here again in the human data, we see that the cycling radioglia are highest in that early one. And later, they actually, the cells that here, the Poliodakis study from Dan Geschwin's lab, the outer radioglia cells are highest. And there's some good expression in the glia too. There's not many glia in this study to compare to. But what's interesting is that this makes it look like, and I'll show you even more data in a second, it makes it look like in the mouse and potentially in the original common ancestor of all these mammals, it is likely that this program existed in the absence of basal or outer radioglial cells, but it existed in gliogenic progenitors. And that program, whether it gave another proliferative push to create more glia or whatever it's doing, it's acting in the neurogenic progenitors in the primates and especially humans to again potentially increase the neurogenic potential of neural progenitors that's required to build the big brain that we have. But originally, that's not what it was for. It was building glia. At least that's what it looks like. So this is another view of very similar concepts, different data sets. This is the Lamano data we've been using to capture these signals, but we have more cells displayed here, gliogenic and other cells that we actually took out because we were interested in the neurogenic trajectory. So this pattern 5 is that original radioglial pattern, here again expressed all over the neurogenic radioglia neural stem cells. The outer radioglial signature here is just on the edge where these pink cells are, which are glioblasts, again showing it as gliogenic NPCs in the mouse that are expressing this. In the human, however, here's the early radioglial signature expressed highest in dividing radioglia. Here, the outer radioglial signature is expressed most highly in here. They did not make any outer radioglial calls in the Trevino data set. This is what they called late radioglia, which is probably a lot enriched in outer radioglial type signatures. And also there is the gliogenic progenitors over here. So it is expressed in gliogenic progenitors in the human, but not specifically so. It's actually peaking in the neurogenic progenitors in the human, where it's clearly peaking in the gliogenic progenitors in the mouse. And I'll skip this one, but this is another study, an earlier one that, again, from the Kriegstein lab that we can see the same kind of effects in. So just to recap, um, our approach is to get all the data we can together, and it's not just so we can do analyses. Everything you've seen, 90% are screen captures from this NeMO analytics site um, that I'll take us to right now and give you a little taste of what we can see there. So this is the landing page for, I don't think I am sharing correctly. Let me share. So I took that link. This is where that link took us. And this is the NeMO Analytics website. This kind of just explains the things I've been telling you about. And this is a sort of a matrix of links. These are different things you can look at, from individual genes to the 7 patterns we looked at, or the 40 patterns we looked at, and some other things like cell cycle genes or disease gene lists. So these are genes that you might want to look at, or gene signatures. And down here are data sets. This is just either the data sets in figure 1 or 2 of our paper, a whole collection of in vivo human data, in vitro human data, organoids and such, in vivo mouse data, in vivo non-human primates, and so on. And then so you pick the collection of data in which you want to either look at a single gene or look at these gene signatures. So for instance, for the 3 data sets we started with, those are in figure 1 of the paper, we can look at the cell cycle gene expression just to get an idea of where things are and give you an idea of what kind of things you can do with NeMO Analytics. All of these links are live in the paper. You can go to the paper and they're all there, or grab them right out of the description of the video. So you can explore the signatures and individual genes as you want. So these are the mouse, monkey, and human data sets we'd started with. As we discussed earlier, you can see in the cell cycle where these actually cells are in the cell cycle by where they are in this loop. This is Nicola Micali's monkey data here. That's the G2M. And if we look at that picture as it changes, looking at S phase, now it's going to come up and it's at a different point, really the sort of on this inside part of each loop in the IPCs up here and the radioglia cells down here. Um, Maybe just one more thing. I'll show you the links to individual genes. This collection of in vivo data, if we hit on that, brings up a single gene in that in vivo collection of data. And a lot of these data sets you will have seen throughout my talk. But here, it's live, so you can look up any gene that you want. So here we're grabbing the E-mesodermin gene, and now it's just going to load a different data set in each one of these. This is more data from the Kriegstein lab, Aparna Baduri in her 2021 aerialization paper. Here is the original study we've been talking a lot from Alex Pollan, also from the Kriegstein lab, where they originally described a molecular characterization of outer radial glia. And here's visualizing E-mesodermin. So this is lighting up intermediate progenitors in each one of these data sets. Um, there's this common theme of a whale and its tail, and those intermediate progenitors are really where the whale and the tail connect in each one of these maps. Um, but again, public, ready to go. You can type in any gene you want. Bulk RNA sequencing across the human lifespan. This is brain span, many regions, many ages. This is from the Lieber Institute, bulk RNA-seq and the DLPFC. And as you can see, there's just a lot of data out here. And this is really our effort to get in the hands of as many people as possible. For people without coding skills, you can type in a gene. For people with coding skills, you can look here. You can download the H5AD for this. If you simply copy this link, you can use a command line wget statement from Unix and grab that data set as is here with the metadata ready to go do any analysis you want on it. So that is really what I wanted to share with you today. I hope it was useful, and I hope you have time to take a peek at the paper. That the link is right here, and I'll put it in the description so you don't have to type all that out. Thanks.